Alcohol consumption has previously been linked to the development of disease. But how does alcohol consumption, especially long-term consumption, as well as binge drinking, impact us genetically? Dr. Lifang Hou, Chief of Cancer Epidemiology and Prevention in the Department of Preventive Medicine, joins me to discuss this question, as well as the link between genetics, lifestyle, and environment when it comes to preventing disease. She recently led a Northwestern Medicine study examining the relationship between alcohol and epigenetic age acceleration. Welcome to the show.
Thank you for having me.
You're an epidemiologist with a multidisciplinary background in medicine and basic science, and a lot of your research is focused on cancer. But your main overarching goal is to understand the link between genetics and environment when it comes to preventing disease. So tell me more about your work.
I am a trained molecular epidemiologist, trained in medicine, basic science, and population science. So my work focuses on finding risk factors in our life, for example, our lifestyle or the environmental, and what biological changes or changes at the molecular level by these risk factors may lead to diseases. So it is important to us to not only understand the causes of diseases, but also the biological changes of the diseases, because before the disease development, these biological changes in our system, for example, in our blood, in our organs, can guide us in disease prevention and can also let us know what are the levels of the risk exposure at the limit. So this will guide the policymakers in policy making and also help the scientists, engineers to develop a diagnostic or screen tools for disease.
Tell me a little more about your background. You were actually a practicing physician for a while, but early on in your career, you pivoted from the clinic to researching populations for causes of disease instead. Tell me about this switch and why it was so necessary in your mind to really further what we know about disease prevention.
Yes, I was more interested in understanding the patterns of the disease, the patterns of the risk factors that may cause diseases more than just treating individual patients. I believed that understanding the patterns or prevalence of certain risk factors and how they cause diseases will make contributions in the prevention of the disease at a large scale. I think it's the main motivation for me pursued further education to get a PhD in epidemiology and human genetics.
So you look broadly at lifestyle and environmental risk factors as potential indicators of disease, but you also look at biomarkers that indicate past exposure as well as future disease risk. So let's drill into this a little bit further. Tell me about these two categories that you study more specifically.
I think they are two sides of the same coin. Exposure to certain risk factors cause some biological changes in human system or organs. For example, in our blood or specific organs in our brain, liver or kidney. So if the exposure continues, the biological changes will accumulate over time in response to the continuous exposure. So biomarker of risk exposure tells us how the exposure may cause a certain change in our system. We call it a biomarker of exposure. However, when the accumulation of certain biological changes reach to a certain point, it will cause some biological or pathological changes that may lead to disease development. So these late-stage biological changes may help us to predict certain diseases. So we aim at developing measurements not just of past exposure, but also its cumulative biological impact that may help physicians, scientists to predict certain diseases. This will in turn help us quantify the risk of getting a disease as a result of that accumulation. So our ability as a researcher to study one or the other is mainly currently dictated by the type of data on which we have access, but in the doctor's office, both could be used to test the blood of healthy individual. In one case, it would give the doctor a more accurate measurement of the patient's health behaviors and their biological impact, which would give them more information to facilitate a discussion of unhealthy behaviors with their patient. In the other, early detection biomarker could serve as an early warning of disease, signaling to the patients and the clinician that it's time for a lifestyle change or more aggressive preventive or interventive measures.
And what we eat, drink, how much we exercise, where we live, how we handle stress, these are just some of the variables that can interact with our genes and influence disease. And these factors also influence both biological age and what's called epigenetic age. So define these terms for me, biological age versus epigenetic age. How are they calculated?
So biological age, we also call it molecular age, is a sort of catch-all term. It can be referred to a wide range of different biological measures that have been associated with aging process in different ways. So epigenetic age is also referred as DNA methylation age. Is just one of the measures of biological age. So epigenetic age uses more about 100 epigenomic changes that have been closely associated with our chronological age. Scientists have found that these epigenetic biomarkers can reflect our environmental exposure history and closely relate to our chronological age. And then we have developed an algorithm. Scientists have found that about 100 epigenomic sites are closely related to human chronological age and turn that into an algorithm, and we referred as epigenetic age. Epigenetic age is designed to be comparable to our chronological age, but the different technology used in the models means that different measures of epigenetic age can tell us different things about what is going on under the hood as we age.
So this specific study that we're talking about today has to do with alcohol. And alcohol consumption has long been understood to compromise health, but its effects on aging are under-researched. Tell us about this study that you led with your colleagues looking at alcohol consumption and epigenetic age acceleration in young adults. What did you find?
We have been very fortunate to have the opportunity to work with the cardiac cohort, which is a very well-established and famous cohort of young people who were enrolled into the study in their early 30s, and they have been followed up for more than 35 years. During the follow-up, they have been examined for various health outcomes, including their subclinical, environmental, lifestyle, and many other outcomes. So alcohol is one of the lifestyle factors that have been studied over time. So the uniqueness of our study really reflects the strength of the cardiac cohort. We have detailed information about the study subjects' alcohol consumption going back decades, adapting the idea of a smoking pack year that measures cumulative behavior of smoking. We also constructed a measure of cumulative alcohol consumption, and we examined how the overtime alcohol consumption may be related to their epigenetic aging process.
And epigenetic aging, you just told us a little bit about. There are actually 4 measures of epigenetic aging in this investigation. Tell me about these different measures and what they mean.
So the fundamental approach to all 4 is similar. All 4 makes use of large statistical model that sift through the hundreds of thousands of methylation or epigenetic sites in human epigenome and look for specific sites. 3 measures of epigenetic age look for sites that are associated with chronological age. One of these, intrinsic epigenetic age, is designed to capture cellular aging. This makes it, in some sense, the purest measure of biological age, independent of environmental factors. The second of these, extrinsic epigenetic age, is designed to capture aging in immune cells specifically and is more sensitive to environmental effects. Finally, there is one also called the pheno age. From the name, you can tell this measure was designed to capture size related to age-related diseases and physical functioning, basically a measure of healthy aging. But the main measure of epigenetic age in our study is Grimm age, which was designed around the methylation sites associated with lifespan, that is time to death.
So let's talk a little bit about what you found in this study. You examined whether cumulative alcohol consumption, which is the number of years that a person consumes beer, liquor, wine, just their total alcohol consumption, as well as the presence of recent binge drinking with these 4 measures that you just described. Tell me about the associations that you found.
So our study found that cumulative alcohol consumption overall and of liquor specifically both influence epigenetic age. On the other hand, binge drinking may only have more short-term effects. Of course, this is only based on our cardiac study. More studies are needed. Additionally, we also observed individuals who are younger in age exhibited greater epigenetic age compared to older individuals, suggesting chronological age in the setting of alcohol consumption may affect aging differently. The picture is less clear concerning beer and wine specifically, but that just means there is more work to do yet.
So this may be concerning for folks that do drink on a somewhat regular basis. Tell me, do you think it's possible to reverse this aging as a result of alcohol consumption? Is this something that you're interested in looking in further?
It is possible, and several studies have identified healthy lifestyle behaviors can result in younger epigenetic age. However, these are mainly observational studies, meaning that they only examined epigenetic age at one time point. What we really want to do is to look the cumulative exposure in relation to epigenetic changes in a prospective way or longitudinal setting to understand the dynamics of epigenetic change. We still have a lot of work to do on whether epigenetic age can be deliberately altered. We will also need clinical trials, and we need to know how quickly epigenetic age can be changed in response to lifestyle changes or any intervention. So additionally, studies exploring whether alcohol cessation will reduce the accelerated epigenetic aging caused by alcohol compared to individuals who continue to drink is an additional area of interest.
Based on what you saw in this study with the cardia participants, would you say that there is an okay amount of alcohol for people to drink that doesn't impact their epigenetic aging?
This is a very difficult question because this is only based on one study. The optimal study design would be conducting relatively healthy individuals to avoid any reverse causality by any disease because epigenetic age can be also accelerated by any existing diseases. Cardia is an ideal study for us to study any risk factors that may affect our biological aging process. However, we also need validations in other studies that are well designed with similar risk exposure measurement and also disease outcomes. So that is what we are working on. Hopefully, we will have some optimal, promising results for us to report back.
What is sort of the consensus in the health community, in the medical community about alcohol consumption? What is safe, or is any amount of alcohol safe?
The results on the effect of alcohol on human health has been conflicting. I think one of the reasons is that we don't have an accurate assessment on the adverse effect of alcohol. More studies and longitudinal studies are needed to really study the long-term effect of alcohol on health on different diseases. this kind of studies in general requires a large cohort, requires decades of follow-up to have sufficient numbers for certain diseases. So this is one of the reasons that so far the results have not been consistent. More studies are needed, and that's one of areas we have been focusing on.
Much of your work has to do with cancer epidemiology. Do these findings correlate to a relationship between alcohol and cancer development as well in this context?
So far, except for liver cancer, alcohol has not been clearly linked with any cancer development. Scientific evidence does show some biological changes induced by alcohol associated with cancer development. More studies are needed. More solid evidence are needed if alcohol really causes cancer or have a carcinogenic role.
Northwestern University Feinberg School of Medicine is very interested in learning more about aging and how to extend life. And this study that was published in the journal Aging is very much connected to that goal. Tell me broadly about some of the work being done at Northwestern to address aging and chronic diseases and chronic disease prevention.
Many scientists in our medical school are studying aging, not only brain aging, but other aging-related diseases. Our group focuses on study how molecular aging can be accurately measured and what are the causes of molecular aging. How molecular aging study can guide us in finding tools or biomarkers that guide the aging disease prevention. One of the aging biomarkers is molecular aging. That's what we have been focusing on. We still have a lot of work to do in addition to our current study on epigenetic aging. So I think it's very important to let the patients know what bad things is happening in their system and how diseases will affect their life that they want to live. So this is our work to find biomarkers that provide some biological information or evidence to show patients or physicians that things are already happening. We need to do something and to stop it before it's too late, meaning that before the disease has developed.
Well, thank you so much for coming on the show today and explaining this important research, and I'm sure there will be more to come.
Thank you for having me. Thank you very much.
You can follow us on Twitter at NUFeinbergMed. Subscribe and ring the bell to hear about the latest groundbreaking research and discoveries. Thanks for listening.